Assalamualaikum warahmatullahi wabarakatuh. My name is FAIZ bin MOHAMAD, and today I'm going to represent you for our group, which is group biology, for our topic, which is the sexual reproductions. So what is sexual reproductions? Well, as you all can know, asexual reproduction is a type of reproduction that does not involve the fusion of gametes or the change of the number of chromosomes. So there are a lot of type of sexual reproduction such as grafting, layering, and so on. But in our topic, we're going to mention a lot about the tissue culture. So my groups are going to present and make a video about it. So stay tuned.
Hello everyone, Syairah here. Today I'll be sharing with you what is tissue culture and what are the types of tissue culture. Tissue culture refers to a method in which fragments of a plant or animal tissue are introduced into a new artificial environment where they continue to function or grow. So while fragments of a tissue are often used, it is important to note that the entire organ are also used for tissue culture purposes. So here such growth media as broth and agar are also used to facilitate the process. Now, let's take a look at what are the types of tissue culture. There are 10 types of tissue culture, but I'm going to explain to you only 4 types of tissue culture. The first one is seed culture. Seed culture is the type of tissue culture used for plants such as orchids. So for this method, eggs plant are obtained from an in vitro derived plant and introduced into an artificial environment where they get to proliferate. The second one is embryo culture. It is the type of tissue culture that involves the isolation of an embryo from a given organism for in vitro growth. So embryo culture may involve the use of a mature embryo, where mature embryos for culture are essentially obtained from ripe seed. So in doing so, the embryo is ultimately able to produce a viable plant. The third one is callus culture. The callus is produced when eggs plant are cultured in an appropriate medium. The last one is organ culture. Organ culture is a type of tissue culture that involves isolating an organ for in vitro growth. So hence, any organ plant can be used as an explant for the culture process, such as shoot, root, leaf, and flower. So there are a number of methods that can be used for organ culture. These include plasma clot method, rough method, agar gel method, and the last one is grid method. So that's all for me. Keep calm and love tissue culture.
Hello everyone, my name is RAFHANAH with matrix number AS12099. Now, I am going to explain the major stages required in plant tissue culture. Plant tissue culture consists of 3 major stages, which are initiation, multiplication, and root formation stage. I will explain briefly each of them. The first stage, initiation stage. This stage is the first phase of tissue culture. Here, once the tissue of chosen plant is obtained, it will be introduced and sterilized in order to prevent any microbes from negatively affecting the process of tissue culture. During this stage, the tissue is initiated into culture. The second stage, multiplication stage. In the second stage, the cultured tissue is redivided and then introduced into the growing medium. Here, the growing medium is composed of all the appropriate components necessary for the growth of the plant, including regulator and also nutrients. This stage is responsible for the proliferation of the tissue and production of multiple shoots. The last stage, root formation. Finally, at this phase, the roots of the cultured tissue, now called as plantlets, are formed. Thus, it is crucial for hormones to be provided in order to induce rooting and consequently complete the plantlets. And there we have it, 3 major stages of plant tissue culture. That's all from me. I'll pass to the next member. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. My name is FAIZ bin MOHAMAD, and today I'm going to present to all of you about the advantages of tissue culture. So let's dive in. So, what is cell potency? Well, cell potency is the cell abilities to differentiate into other cell types. The more cell types a cell can differentiate into, the greater the potency. Let me explain to you the advantage of it. So, we're going to continue about the advantage of tissue culture. So, for the point number 1, I'm going to say that tissue culture advantages, which is any plant can be freely from any disease or viruses. Why it can be freely? Obviously, cell potency has already differentiated this gene and altered it to become immune from any disease or viruses. Hence, it will increase the resistance to it, any virus or bacteria, so that it will become more tough from any other normal plants. For the second point, which is this plant is not dependent to seasons, and you can obviously plant it whenever you want, as long as you have a few basic types that require for a plant to grow, which is sun, wind, oxygen, water, and nutrients. Sufficient nutrients, obviously. So there is my 2 points advantages for tissue culture. Let's move on to another point. So for the last point, I want to ask you, have you ever wondered how a farmer can grow their plants such a very short amount of time and make such a more profitable income by selling their own plants? Well, that is one of the advantages of tissue culture. Why? Because tissue culture can also grow their plants in such a short amount of time, because cell differentiation can occur in such a short amount of time and makes the plant grow really well. So I think I'm gonna wrap up these vlogs by saying that there's a lot of advantages for tissue cultures that our society can also implant it in our daily life. So this is all from me. Thank you very much.
Hi, Assalamualaikum warahmatullahi wabarakatuh. My name is MOHD and for today I'm going to tell you guys about technique for plant in vitro culture. So there are 3 techniques that are often used, which is somatic embryogenesis, meristem tissue culture, and anti-tissue culture. So for the first one, I'm going to talk about the somatic embryogenesis. It is defined as the development of an embryo derived from somatic cells under an appropriate artificial environment, which is in vitro itself. It is a significant process of the tissue culture as it offers a rapid large-scale propagation system that is useful for many industries. And there are 2 types of somatic embryogenesis, which is primary somatic embryogenesis. In this type, the embryogenesis can only be induced by using the explant. And the second one is secondary somatic embryogenesis. In this phenomenon, the development of embryo is induced through existing somatic embryos. Now, the second technique is meristem tissue culture. So meristem tissue culture involved in the removal and in vitro placement of dome-shaped meristem tissue culture on the culture media for the development of whole plant. It become popular among culturists mainly to obtain pathogen-free plant. There are 4 meristem tissue culture application. The first one is virus elimination, second mass propagation, third germplasm storage, and the last one is genetic transformation. And the last technique that I will be talking about is anti-tissue culture. So anti-tissue culture is used to develop instant homozygous inbred lines, avoiding the lengthy time needed using traditional SYAFIQ method, including bud pollination. Anti-culture result in haploid plants, and it can easily identify by their smaller sterile flowers. So for the next session, it will be the interview session. So stay tuned.
So in meristem culture, the virus cannot go to this meristem part because meristem part doesn't have vascular bundle. Virus spreading in plants through this vascular bundle, through xylem and phloem. So only when we want to get virus-free plantlets. So if I want to give example for anther or pollen culture, because anther is female and pollen is male. So when you cross chili A with chili B, you will get a variation of results. For meristem culture, the technique usually used to eradicate virus, and for anther culture, usually we want to produce purebred line. So we are not talking about different plants. We are talking about what we want to do with our cultures, with our plants. So let's say, other than meristem culture, other than enter culture, we have so many other techniques. We have shoot tips culture, we have nodal culture, we have meristem culture, enter culture, somatic embryogenesis. So the techniques is so many. It's just that what is our objective? When you do micropropagation, you will produce mass of plantlet. When we get numbers very high in numbers and they are identical, we call them as clones. Micropropagation produce clones, but they are not cloning technique. Or we have fungus infection inside the plant already. They are endogenous. What we need to do, if we know the plant is containing containing fungus or bacteria, is we add onto the media antibiotic. The bacteria or the fungi inside the plants, they will get eradicated. My answer is always, if you can do it yourself, why not? First is pressure cooker. Secondly is that laminar airflow. You can substitute laminar airflow with that aquarium. And then thirdly is, you can manipulate media. So what we can do is buy fertilizers sometimes and then mix and then dissolve that fertilizers, and then you just mix and match. Sometimes you will get a DIY media.
Okay, first of all, it depending on how good you are to handling the technique practically in the lab. So before starting any work, for my practicing also, actually I will wash my hand first. I will wash properly, starting from my fingers until my elbow with using the soap. So after that, I will wear the mask, the lab coat, and also the glove. So next step, please make sure all your equipment will already sterilized, like forceps, petri dish, bottle, and nutrient medium that used to culture that sample. So moreover, the best part or the better part is to make sure the mother plants that you choose is in healthy condition without any damage or no disease. So that will make your tissue culture successfully without any contaminated bacteria. In my opinion, there is two answers. There is an impact and there is also no impact. Okay, there is an impact actually for ornamental plants lah. So people now rather saving money for buying food compared to ornamental, buying plants ornamental. But for no impact COVID-19, kalau pokok yang mendatangkan dan boleh buatkan makanan macam like papaya, banana, I think there is still demanding on that, and no issue, no big issue COVID-19. Sebab petani pun masih mengusahakan makanan untuk support keperluan negara. Jadi untuk tissue culture in this area boleh dikatakan ada dua lah. Kalau untuk ornamental plants tu mungkin akan slow sikit lah dia punya sektor. Tapi kalau untuk makanan rasanya tak ada big issue lah. Mungkin masih selamat lagi. Sebab manusia masih mencari makanan kan. Berbanding hiasan sekarang lagi. Sebab duit lebih penting digunakan untuk membelikan makanan sekarang. Itu je pendapat saya.
So in conclusion, there are many benefits that human can get from the tissue culture techniques, such as the faster ways to grow up our plant, and the plant that produce are also disease free. So I think that's all from our group, and thank you for watching.